Welcome to another interesting topic of botany of molecular biology, that is translation. Now we already have discussed the different molecular biology aspects like DNA replication, RNA transcription. Now next is the process of translation. So what do you mean by translation? Now it is the process in which the protein is synthesized from an mRNA transcript or from an mRNA copy. So how that process occurs, that how that mRNA codons or the nucleotides present on the mRNA, it codes for certain kind of amino acid, and how the amino acids are joined together to form a long chain of polypeptide, and further after certain modification it becomes a functional protein. That is the theme of this chapter today. So before going to translation in details, now again let's focus on the central dogma of molecular biology. We already have discussed that the central dogma of molecular biology says that from a segment of DNA or from a gene, RNA transcripts are formed by the process of transcription, and from only the mRNA copy or the mRNA template, now polypeptides are synthesized in the process of translation. But there is a big question, that how the nitrogenous bases or the base sequence or the nucleotide sequence present on the DNA or that is being transcribed into the mRNA, and how this nucleotide base sequences, how they are being transformed to the amino acids, or how they can direct the synthesis of a polypeptide, that is a big question mark. And this question is answered by another chapter or by another concept, that is genetic code. So before going to translation, we will discuss the process of protein synthesis by the concept of genetic code, and exactly how the translation process occurs. So genetic code, regarding genetic code, let me give you a very few introduction, that from a DNA an mRNA copy is produced, and from the mRNA now a polypeptide or a protein is produced. So this is something like one gene, one polypeptide hypothesis being said by or being proposed by Beedham and Tata, and this process is known as transcription, and this is translation. So when it comes to translation, the question is how the nucleotide sequence present on the mRNA, how does it help in conforming the synthesis of a polypeptide. That means now if this is a stretch of mRNA from 5 prime to 3 prime, now the base sequences that are present over here, how they are being converted to a sequence of amino acids. Okay, how the nitrogenous base sequence or the nucleotide sequence, they are converted into an amino acid sequence being stringed within a polypeptide, and further it forms a functional protein. So this question is answered by the concept of genetic code. Now we have 4 nitrogenous bases. So as we may take as adenosine phosphate as a nucleotide, a guanosine phosphate, a cytidine phosphate, or a thymidine phosphate in DNA, or a uridine phosphate in the case of mRNA. Now whatever it may be, now if we take that a single base codes for a single amino acid, now we will get 4 amino acids. but in true or in the true sense, now we have 20 amino acids in our cell. So inside a cell we have 20 different amino acids. So how they can be coded? By this 4 nitrogenous bases only. So let's take a different combination, like let's take 2 bases at a time. So if we are taking 2 bases at a time, now we are going to get 16 amino acids total. So 16 amino acids cannot be it cannot fulfill the total 20 amino acids. So next concept is that it should be having 3 nitrogenous bases or 3 nucleotide sequence at one time. So if we are taking that the code is a 3 nitrogenous base sequence, then it may code for 64 codons maximum. So the 64 codons easily can code for 20 amino acids. So that's why now we have defined the genetic code as the genetic code is a set of 3 letter combinations of nucleotides called codons, and each of which corresponds to a specific amino acid or stop signal. Once again, genetic code is a set of 3 letter combinations of nucleotides called codons, each of which corresponds to a specific amino acids or a stop signal. Okay, so in total we have 64 codons. How that will, I'll tell you. So in total we have 64 codons, and it is in the form of triplet. So the codons are triplet in nature. That means minimally 3 nitrogenous bases or 3 nucleotides are required for coding for a single amino acid. So the genetic code is a set of rules that define how the 4-letter code of DNA is translated into the 20-letter code of amino acids, which are the building blocks of proteins. Once again, now we have 20 amino acids inside a cell, and there are only 4 nucleotides or 4 nitrogenous base. Now this is the nitrogenous base sequence that determines the concept of gene, the template of mRNA, and the kind of amino acid that has to be added into a polypeptide chain. So this is only the nitrogenous base sequence. Now in that sense, we only have 4 nitrogenous bases. So how this 4 nitrogenous base code is converted to a 20 amino acid code in the process of translation, that is being answered by the concept of genetic code. And the genetic code says that the codon, that means a single codon, it codes for a specific amino acid or a stop signal. Now a single codon is triplet in nature. That means a single codon comprises of 3 nitrogenous base sequence. Well, so we will discuss that in different or in the upcoming slides. So next, let's go to the history. The discovery of codon was first described by Francis Crick in the year 1961. Now during the same year, Nirenberg, Methy, and they performed an experiment that began with disappearing the genetic code. They showed that a RNA sequence having the U bases only, okay, or the uridine nucleotides only, now they are coding for what? Phenylalanine. Well, so in what sequence? Now let us suppose there are 30 U residues. Then it is coding for 10 phenylalanine in a polypeptide chain, and 10 phenylalanines are present within that polypeptide. So in this way, they confirmed that triple U, it codes for phenylalanine. Further, other scientists or same the Nirenberg, Leader and Hargobind Khorana, they identified the rest of the genetic code and this fully described each 3-letter codon and its corresponding amino acid. So what they did, that they have taken a permutation combination of many the nitrogenous bases, and they confirmed or they concluded that this 3-letter sequence of nitrogenous bases, it codes for this particular amino acids. Well, so this is the historical background. Now next, the most important thing is the characteristic feature of genetic code. Now in genetic code, if we will consider the genetic code, now as I told you, now if we will take only 4 nitro, or if we will take that a single nitrogenous base codes for a single amino acid. Now how many we are going to get? So we are going to get A, T, G, C. Now this will code for how many? This will code for 4 amino acids only. If we will take it in doublet like A, T, G, C, this is in case of DNA, we may take U in case of mRNA copy. Okay, so then how many combinations are possible? You just see. So again A, T, G, C, and again A, T, G, C. So if we will count these numbers, they will come as 16. So if we are taking a single nitrogenous base to code or to code for a particular amino acid, we are getting 4 amino acids in total. If we are taking a doublet codon, now we are getting 16 amino acids in total, but it is not satisfactory that it codes for all the 20 amino acids. So next phase is that we will consider that this genetic code is triplet in nature, something like this, or it may be something like this, or it may be something like this and all. Okay, so what we are going to take, so we will take it as something like this, A, T, C, G, or you may take it as U. Then again this branch will take A, U, G, C. Okay, then again this will with 4, this will with 4, like this. So each one getting 16, 16 into 4, 64. So here this is the tabular form where we are getting all the codons in triplets, okay, and they code for a specific amino acid. Well, so coming to the features of genetic code, now we have 64 codons as I showed you. So here we have in total 64 codons. So when we are taking triplet codon in nature, and all the codons are triplet in nature and they codes, or there are totally 64 codons. So out of this 64 possible amino acid combinations or permutations, 3-letter nucleotide sequences that can made from 4 nucleotides. Now out of the 64 codons, 61 they codes for specific amino acid, and the rest 3 that doesn't code for any amino acid. Rather, they are considered as stop signal or stop codons or termination codons. And this termination codons, this is specifically UAA, UAG and UGA. These 3 are the stop codons. Well, that doesn't code for any amino acid. Now, although each codon is specific for only one amino acid or stop signal, still then some amino acids are represented or they are being coded by more than one codon, and that's why the genetic code is degenerate in nature, or it is called as redundant. Okay, so the degenerate nature of genetic code explains that many amino acids are there which can be coded by more than one codon, and this explains the degenerate nature of genetic code, and this has been best explained by a hypothesis known as Ubel's hypothesis. And the Ubel hypothesis says that this is only the interlocking between the codon of mRNA and the anticodon present on tRNA, and because of there is a loose kind of interaction between the third base of the mRNA and the first base of the tRNA, there is some kind of interaction and many of the bases can be changed at that particular position. So, the genetic code is degenerate in nature. Now, let us see how it is degenerate. Now, you see here an amino acid called as serine. Now, it is being coded by 4 triplet codons like UCU, UCC, UCA and UCG. Similarly, many things are there. Now, you see the stop codons like UAA, UAG and UGA. Then here you can see arginine is being coded by 4 triplet codons like CGU, CGC, CGA and CGG. So, many such examples are there where a single amino acid is coded by more than 1 codons. So, what are the next features? The next important features are the genetic code is commaless, it is non-overlapping, and most importantly it is nearly universal. That means it is well applicable to all kind of organism starting from Monera to the kingdom Animalia. Well, so this is all about the characteristic features of genetic code, and this is the genetic code table which explains that the different amino acids is being coded by the different kind of codons. In total there are 64, and this is the genetic code. Now, you see from the DNA segment, now this is an mRNA is being produced, but here the T should not be there, rather there be U, this is also U, and the rest of the things are same. Now, if this is a copy of mRNA, now this is like AUG, it codes for 1 amino acids and the amino acid is methionine. So, likewise this is CUA, this codes for another amino acid, GGC, this code for another amino acid, and likewise the different triplet codons, they codes for a particular kind of amino acids and they are being joined together to form a polypeptide. So, this is the process of translation, and it is being covered on the background of the genetic code. Okay. So, this is all about the genetic code, how it explains that how translation occurs from an mRNA transcript to a polypeptide chain. Okay. So, next we will go to translations directly. Now, what does it mean by translation? Translation refers to the process of polymerization of amino acids to form a polypeptide. Once again, let me tell you, whether it is replication, transcription or translation, all the 3 are polymerization process. Now, in case of DNA replication, DNA, it is a polymer of nucleotide, particularly deoxyribonucleotides. In case of transcription, this is polymerization of ribonucleotides, and here in case of translation, it is the polymerization of what amino acids, okay, into a polypeptide chain. So, the order and the sequence of amino acids are defined by the sequence of bases in the mRNA. As we already have discussed this is the base sequence of mRNA which actually decides that what kind of amino acids will be joined together one after another in a sequence. So, it is determined by the base sequence that are present on the mRNA strand, and the amino acids are joined together by a bond known as peptide bond. So, as I told you that 2 nucleotides are joined together by a phosphodiester bond. Similarly, 2 amino acids, they are joined together by a bond known as peptide bond. So, if I will just draw a string here, now the beads are being the amino acids. So, these bonds are the peptide bonds, and it also has 2 ends. So, one is a amino group end, the other one is a carboxylic group end. This is because of the structure of amino acid. So, let me show you the structure of amino acids. Okay, so what is a peptide bond? Now this is regarding the structure of amino acid. Well, so later I'll show you the structure of amino acid. Now this is the peptide bond which holds together two amino acids. So which holds together two amino acids. Now regarding this, if I will draw the structure of amino acid, now this is the structure. Now this is a centrally placed carbon which is being bonded by four different groups. Okay. Now there is a carboxylic group, an amino group and a hydrogen atom which are being attached to the central carbon, and the rest is called as a functional R group. And this functional R group, it differs from one amino acid to another amino acid. Now when the functional R group is an H atom, it refers to glycine. So when it comes to the structure of glycine, now this R group is a H atom only. Now when it becomes a methyl group like this, now this becomes the structure of alanine. So likewise we have different functional R groups for the 20 different amino acids. Okay, so all the 20 different amino acids have different functional groups, but the common thing is that they have an amino group, a carboxylic group and a H atom. Now next, how they form a peptide bond. So the peptide bond is formed between two amino acids. So one is this amino group, this is the carboxylic group. Let us suppose this is the first amino acid 1. Then this is the second amino acid. This is amino group or amino acid 2. Now with the removal of a water molecule, a bond will be formed, and this bond will be something like this, and this bond is called as a peptide bond. So in this way, after this another amino acids come and join, it forms another peptide bond. Again with another amino acid, it comes and joins, another peptide bond is formed, and likewise the peptide bonds are being formed. So let's go to the slide. Okay, so let's go to the slides. Now before the actual act of translation, there should be charging of this amino acid. So there should be charging of tRNA or amino acetylation of tRNA. That means now we already have discussed the structure of different RNAs like mRNA, tRNA and rRNA in the last class. Now if you can remember the structure of tRNA, there is a point where the tRNA carries the amino acids, and there is a point called as anticodon loop where the anticodon and codon interlocking happens. So the tRNA, before it interlocks with the codon of the mRNA, it should carry an amino acid. So everything is there in the cytoplasmic pool. The amino acid is there, the tRNA is there, but they should be joined together as per their identity. They should join together. That means a particular kind of tRNA should carry a particular kind of amino acid. So this point is called a charging. Let us suppose there is a tRNA which has to carry the methionine. So there is a point called as, or there is a tRNA called as initiator tRNA which has to carry an initiator methionine, that is a formulated methionine. So then together the joining of the tRNA or initiator tRNA with a formulated methionine, it is called as charging of that initiator tRNA. So charging means loading of the amino acid to the tRNA is called charging of tRNA or amino acylation of tRNA, and this is the prior requirement for the act of translation. If charged tRNAs are brought together very close to each other, then only a peptide bond will be formed between two consecutive amino acids, and it is being affected by the ribosomes or by the catalytic activity of ribosome, which is called as peptidyl transferase. So next, a translation unit. Now let us suppose there will be this stretch of mRNA which has to be translated. Now it does not mean that the whole length of mRNA will be translated into a polypeptide. Rather, there is a specific reason which should be translated into a polypeptide. So that reason which only will be translated, it is called as open reading frame or a translation unit, or it is called as a system. So this is a unit of function. Okay. Now this open reading frame, it starts with a initiation codon that is called as AUG, and it stops at a stop codon or a termination codon. As I have already told you that the stop codon can be UAA, UAG or UGA. So out of these three, any codon can be a stop codon. So within this flanking region, whatever would be there, it will be translated into a chain of polypeptide, but not the region which are prior to the initiation codon and later to the stop codon. So that means these regions, they will not be translated. So the region between initiation codon to stop codon, this is called as a translation unit and the other portions of the mRNA, they are called as untranslated region. So this is the translated region TR, and these are the UTR, untranslated regions. So a translation unit in an mRNA is the sequence of RNA that is flanked by a start codon and the stop codon and codes for a polypeptide. And mRNA also has some additional sequence that are not translated, and these are referred as untranslated regions, as I told you. So the untranslated regions are present at both 5' end and 3' end, before the start codon and after the stop codon. So this is all about the region where your translation occurs only. So after activation of amino acid, your translation process occurs in 3 steps. One is initiation, elongation and termination. So first let us start with activation of amino acid, how an amino acid is activated. Now the activation of amino acid takes place in cytosol, no doubt, obviously, because both the amino acid and the tRNAs, they are present in the cytoplasm and togetherly they has to be charged. So the tRNA is charged because of the loading of amino acid, and it is affected by an enzyme called as amino acyl tRNA synthetases. Well, so the activation of amino acid is catalyzed by their amino acyl tRNA synthetase. Now this is very specific in choosing the particular kind of amino acid with that particular kind of tRNA. Now let us suppose a tRNA has to load a methionine. So this synthetase enzyme, it decides that which tRNA it has to pick up and it has to join it with the respective kind of amino acid. So it won't join a particular, or let us suppose it would not be going to join a particular kind of tRNA which has to load methionine with that of phenylalanine. So it is very specific in its action. Now all the 20 amino acids are activated and bound to the 3' end of their specific tRNA in the presence of ATP and magnesium ion. As I told you that if you can remember the structure of tRNA, now tRNA holds the amino acid where? Towards the 3' end of the tRNA, where there is a CCAN, and that 3' end adenosine, that nucleotide, it carries the particular amino acid. So, and at that point only it is being loaded. The N-formulated methionine is chain initiating amino acid. Now, in case, now we know that there is a initiation codon, and always the initiation codon is AUG, whereas 3 stop codons are there like UAA, UAG and UGA. But when it comes to initiation codon, it is always AUG, and AUG codes for methionine, whereas stop codon doesn't code for any amino acid, and the initiation codon, it always codes for methionine. So, the first methionine in case of prokaryotes, it has to be formulated. That means there is an addition of a formyl group to the methionine before it is being charged onto a tRNA. So, the first tRNA is called as the initiator tRNA, which has to come and to bind to the first codon, that is AUG, and that initiator tRNA always carries an initiator methionine, which is a formulated methionine. So, it says the N-formulated methionine is the chain initiating amino acid. So, that means always when you consider a polypeptide, in the first or in the polypeptide chain, you will get the first amino acid as a formulated N-formulated methionine. So, methionine is the initiator amino acid. So, is the chain initiating amino acid in bacteria, whereas methionine, simply methionine, is the chain initiating amino acid in case of eukaryotes. Methionine is activated by methionine tRNA synthetase. As I told you, a particular kind of synthetase enzyme is responsible for joining together a particular kind of tRNA with its respective kind of amino acid. So, here the synthetase enzyme which will join the tRNA of methionine with a methionine amino acid is called as methionine tRNA synthetase. For N-formyl methionine, 2 types of tRNAs are used. One is tRNA methionine and another one is tRNA F-met methionine. Well, so this tRNA methionine, it may add a methionine at any point between the initiation codon to the termination codon, but this tRNA F-met, it always adds the methionine at the initial point of translation. That means to the initiation codon only. Similarly, all 20 amino acids are activated by amino acyl AMP enzyme complex and then bound to their specific tRNA, forming amino acyl tRNA. So, first of all, let me show you how the tRNA is charged with the amino acid. So, it is a 3-step process. Now, you see amino acid activation is a 3-step process. So, first of all, the amino acid joins with the ATP molecule. It forms an AMP complex. So, it is amino acyl AMP complex with the release of a pyrophosphate. Then, the amino acid is transferred to the enzyme. To the enzyme means amino acyl tRNA synthetase enzyme. So, this AMP amino acid is transferred to the enzyme. So, this is the enzyme you see. Well, and this is transferred where the amino acid is, it remains within the catalytic site of the enzyme and the AMP, it comes out. Why the ATP is being used here? Now, in order to give activation energy or in order to activate the amino acid for the act of peptide bonds formation, now here the energy is utilized from the very beginning by ATP molecule by forming a AMP amino acyl complex. And here the pyrophosphate is released, and this pyrophosphate also provides some amount of energy or some energy which helps in carrying out the whole process. So once the enzyme amino acid complex is formed, then the tRNA molecules comes into the catalytic site, and the tRNA now takes over the amino acid unto itself, and the enzyme is freed now, or the enzyme frees the tRNA amino acid complex. So in this way, the tRNA is loaded with the amino acid, or it is being activated with the amino acid. So this is called as activation of amino acid or charging of tRNA. So it has two ways: activation of amino acid or charging of tRNAs. Okay, so it is a three-step process. So once again, first the ATP, it forms a complex with the amino acid in the form of AMP amino acid complex. A pyrophosphate is released and it provides the energy for joining of this AMP to the amino acids. Then the enzyme takes over here. Now the AMP amino acid, it comes into the catalytic site of the enzyme. Here the AMP is released, the amino acid remains there. Then the tRNA comes into the site and it takes over the amino acid group into itself, and it loaded itself with the amino acid group, whereas the enzyme frees everything with the formation of an amino acid tRNA complex. So this is a clear picture, you can see. Now this is the tRNA. Now here towards the 3' end there will be a hydroxylic group which will hold the amino acid group. Now here you see any kind of, if it is methionine or whatever it may be, now any amino acid, it is being carried over this 3' end, okay, and particularly on the adenosine nucleotide. So this is the charging of tRNA or activation of amino acid. Activation of amino acid means amino acid is neutral in nature, but this activation by the help of this AMP or by the help of ATP molecules, now the amino acid is so activated that it can form the peptide bond by taking the energy from the ATP previously. Well, so next go to the initiation. Now initiation occurs because of all the 3 RNAs. Now for the act of initiation we require mRNA, we require tRNA, the respective tRNAs having the amino acids which are going to form an interlocking between the codon of mRNA and the anticodon of that particular tRNA, and the rRNA of course, but rRNA being a part of the ribosome because the rRNAs are present in the ribosome. So smaller ribosomal subunit, it has a single rRNA, and the larger ribosomal subunit, as per the prokaryotes and eukaryotes, they have 2 rRNAs or 3 rRNAs respectively. So what happens? Let's read out. In the first step, the initiation factor IF3 binds to the 30S ribosomal subunit. Then it says that it is not that the 2 of the ribosomal units, they are joined together from the very beginning. Rather, the smaller ribosomal subunit, it first of all binds to the mRNA strand, after which next the larger ribosomal subunit comes and bind, and now it forms the translation initiation complex. So prior to that, let's read out what are the steps that are being involved in initiation. Then mRNA binds to the 30S ribosomal subunit in such a way that AUG codon lie on the peptidyl site and the second codon lies on the amino acid site. I will show you in the diagram. The tRNA carrying formulated methionine is placed at P site. This specificity is induced by an initiation factor that is called as IF2. With the utilization of ZTP, the IF1 prevents binding of F-met tRNA is in A site. Okay, so the IF1 prevents the binding of this initiated tRNA to the A site or the amino acid site. What are those sites? I will tell you. But the importance being there is a sequence which is present towards the 5' end of the untranslated region of the mRNA, and this sequence is called as SARDAR sequence, and because of this sequence the 30S ribosomal subunit, it helps in binding of this 30S ribosomal subunit to the mRNA template in such a way that it includes the initiation codon, and it lies at the P site, and the second codon at the A site. So this sequence, SARDAR sequence, this is an important sequence which is purine rich, and it helps or it facilitates the act of initiation of translation. So its sequence in the mRNA, it guides correct positioning of AUG codon at P site of 30S ribosome. After binding of this F-mate tRNA F-mate on P site, the IF3, IF2 and IF1 are released so that 50S ribosomal subunit comes and bind to the 30S, forming the 70S ribosome, and the exit site is created. Okay, so now this becomes a translation initiation complex. So let's have a look on the diagram. Now here, if this is the mRNA template, now this is the start codon. So this is the translation region which starts from start codon and ends somewhere at the stop codon. But prior to that, in the untranslated region, there is a sequence called as SARDAR sequence, and this SARDAR sequence is a purine rich sequence where it facilitates the binding of the 30S ribosomal subunit. Why it is so? Because in the 30S ribosomal subunit there is 16S rRNA, and this 16S rRNA joins, or it has a complementary sequence within it which can form hydrogen bonding with this SARDAR sequence, and it helps in such a way that the smaller ribosomal subunit will be placed in such a manner that it will include the AUG or the start codon at the P site and the second codon at the A site. Okay, so how the sites are created now you see here. Well, so first of all, when it is 30S ribosome it is binding, we are getting only P site and A site. But next, when this, with the release of all the factors like IF1, IF2 and IF3, so when these three factors are released from the site, well, now next is that the 50S or the larger ribosomal subunit, it comes and binds to the 30S ribosomal subunit, forming a complete 70S ribosome. Okay, and this becomes the initiation complex. Okay, and the initiation complex carries an E site, P site and A site with a formulated methionine placed because of a initiator formulated methionine tRNA. So there is a tRNA present here which has an anticodon UAC which makes an interlocking with the first codon, that is AUG, and now it forms the initiation complex. Well, so what are this P, A and E site? The A site is called as amino acyl site. So what is that? This amino acyl site, always when the ribosome slides over one codon and one codon at one time, now all the time a fresh tRNA is it comes to the A site. So always a fresh tRNA comes and bind to the A site, which is called as amino acyl site. Whereas the P site, it is called as the peptidyl site. Peptidyl site means in the first instance it is the AUG codon which makes the P site, but later on the peptide chain, it is carried only at this P site. So that's why it is called as peptidyl site, as it carries a long chain of peptide. So always a fresh tRNA comes and bind to the amino acyl site, whereas the P site, it always carries a long chain of peptide, and rest the E site, that means where the tRNA which is present here, it doesn't carry any amino acid. Whatever amino acid would be there, it would have been given to the P site, and here as it doesn't have any amino acid, now it is empty, and this site is called as what? Exit site. Now I think now it's clear. 3 sites are there within the 70S ribosome. One is A site from the right. This is amino acid site. Always a fresh tRNA carrying a single amino acid comes and bind to this site. Then a peptidyl site. This is the site at which a peptidyl chain is being carried. But in the first instance, the AUG codon or the first codon, it makes the P site and it carries a formulated methionine at that site. But later on, with this elongation of the peptidyl chain, now the peptide bond or this peptide chain, polypeptide chain is carried over this P site, and this E site where the tRNA having no amino acid or peptidyl chain, now it comes out of this site. Sorry. So how it occurs? It occurs by 3 initiation factors. One is IF1, IF2 and IF3. Now all these 3 togetherly, it forms the initiation 70S initiation complex. So what happens here? See, now first of all, the IF3 comes and bind to the 30S ribosomal subunit, and it occupies a position where at the A site, so that the A site is blocked and no tRNA can bind to that position. Now next, the IF2 factor, now it comes as the initiator tRNA and the IF2. Now it comes and bind to this side organ or to the initiation codon site, and it places the initiator tRNA at the AUG codon exactly. Now rest, the IF1 comes and bind and it triggers the release of all the factors, and finally the 50S ribosomal subunit comes and bind, and that creates the 70S initiation complex. Now this is the initiation complex. Now you see all the factors IF1, IF3 and IF2 is always being associated with the tRNA. So this is the GTP molecule which is being triggered to form or to release all the factors. After this release, the 50S will bind and it will form the 70S initiation complex. Next is elongation. Now elongation takes place in 3 steps. One is binding of the amino acid tRNA at the A site, and next is the peptide bond formation, and finally there will be ribosomal translocation. So elongation, here the binding of amino acid tRNA at A site. Now what happens here? The second tRNA carrying the next amino acid would always bind to the A site, and the A site is always being mentioned or it is always being maintained at the free site for binding of a fresh tRNA. So what happens here? It is being held by or facilitated by EFTU factor, and it utilizes a GTP molecule. Okay, and after binding, the GTP is hydrolyzed and EFTU GDP is released, and this GDP then enter into EFTS cycle to form another molecule of EFTU and GTP. So next you see, now here the first tRNA is being positioned. Now the second tRNA has to come off with the loading of an amino acid. So it has to come to the A site. Now it's clear. Now A site will always carry or it will always have the fresh tRNA carrying the amino acid. So here you see, now after this, now here you see a dipeptide is formed, and this marks the elongation's first step. And the elongation is a process where it remains in cycle. Okay, so first of all there will be incoming of a fresh tRNA to the A site, then there will be formation of a peptide bond, then next there will be translocation of the ribosome. So what happens here, you see, now here the first AUG or the tRNA, that has the methionine. Now this methionine is transferred to the second codon where it has created a dipeptide comprising of a tryptophan and a methionine. Okay. So next another, now if this is the third codon, now next another tRNA will come to this site, and here this will be a phenylalanine. Then what will happen, this dipeptide will be they will be transferred to this A site, so forming a tripeptide. So each time there will be transformation of the P site to the E site and the A site to the P site. Okay. So this is the peptide bond formation. Then the peptide bond is formed between these two a consecutive two codons, and here the tRNA is carrying the amino acids. Between them a peptide bond is formed. Now here you see, now this carries a dipeptide, and all the time this peptidyl group is transformed. This peptidyl group is transferred to the newly incoming tRNA present on the A site. So what happens here next is, now you see this is a dipeptide that is being formed in the elongation. Now what happens here next, the ribosome translocate. Translocate means it slides over a single codon, and that single codon again it transforms. Now you see it transforms the P site to E site in this way. So the P site is converted to E and the A site is converted to P, and a freshly A site is always being created to carry what, the next incoming tRNA as per the codon anticodon interlocking. So this is about the elongation. In the next class we will conclude the termination. So have a good day and nice day.